today we are very privileged to have with us Professor Ulrich, who will be giving a talk entitled From Single Molecule to Covid-19 Detection, How Physics Can Be Used to Create Novel Tests for Viruses. Professor Kaiser is currently a professor in applied physics at the Cavendish Laboratory at the University of Cambridge, and also a fellow at Granville and Keyes College. Today, Professor Kaiser will discuss his group's recent developments to detect and localize structures as accurately as possible along DNA molecules approaching super-resolution microscopy. The localization is enabled by quantitative understanding of polymer dynamics in glass nanopores, which enables it to extract positional information to a few 10 base pairs. He will also discuss how they apply knowledge of single molecules and nanopores to create novel tests for viruses and their variants that may play a role in the prevention of future pandemics. With that, can we welcome Professor Kaiser, please.
Thank you very much. I hope it works with the microphone. If it's a bit too loud, let me know, because it seems very sensitive. Okay, so I would like to thank the organizers to give me the opportunity to talk a little bit about our work. And when he read out the abstract I submitted, I noticed that I cut out probably 60% of that stuff and will just focus on something that I think we are recently really excited about. And this is basically general test for viruses, but I will also try to motivate the biophysical aspects for this, how actually really going from single molecules you can really do something maybe that is possible for the relevant for the real world. And I know that I wrote down here COVID-19, and for people who follow this of course know that this is the disease, not the virus. Yeah? I put this down deliberately as COVID-19 because we are actually not very our test is not very useful for early detection or very sensitive, but more for diagnosing people who actually have already symptoms and maybe can go to the clinic, but again I will come back to this a bit later. Okay, so I wanted to also give you an opportunity, use the opportunity to tell you what we are doing in my lab in general. So I'm working on 4 different areas. There's a group of around 15 PhD students and postdocs, and first of all, this is the physics of channels. So this is really the physical basis for all of the work we are doing. This is statistical physics, where we try to understand movement of small particles in confined geometries. So this is thermodynamics and statistical mechanics on the micron scale. So we call this mesoscale because you can still see these particles. Then we have a large effort on building this DNA, and this is the basis for the test that we are developing for the SARS-CoV-2 virus. It is relevant for the COVID-19 pandemic, and here I will talk about more today and will introduce you to the ideas here of DNA nanotechnology, because I think this is really a nice area to understand where there's an intersection between the life sciences and the physical sciences. The other basis for our detection of viruses or viral genomes, to be more accurate, is nanopore sensing, and this is another maybe also interesting item in the news to also understand a little bit more, because I don't know how many of you follow stock markets, but there was recently a share issue by Oxford Nanopore technology from the other place for a new DNA sequencing technology. And I should also have said that actually part of this work is actually funded by companies. Yeah, and let me just go back here. So you see here Oxford Nanopore is one of the sponsors of my lab. So this is basically the 2 things that I will combine in this talk. The last area that is kind of connected to all 3 of them is the membrane transport. I won't have time to talk about this at the moment, but here, of course, we are also looking at drug transport, peptides going through lipid membranes, other small molecule transport, but again, this is not the focus of the talk today. So, I want to, if I have one aim of this talk, is that I want you to get home and remember that fluctuation and randomness in our bodies and on the micro and nanoscale is absolutely dominating and extremely important. Yeah, so what you see here is an illustration of this. This is actually a microfluidic chip with so this barrier here that you can see is around 10 micrometers across. So 10 micrometers is roughly a fifth or a tenth of a human hair. So depending on how thick your hair is, you see my hair is very thin, so it's probably more on fits if I have any hair left. But so for young people or for people with thicker hair, hair is roughly 100 micrometers. So this barrier here is actually 10 times thinner than human hair. And this channel is 1 micrometer across roughly and 800 nanometers high. So this is 100 times thinner than human hair. And these particles that you're seeing diffusing around, this is by the way real time, are around 500 nanometers in diameter. So 500 nanometers in diameter is around 250 times larger than the DNA molecules that we are using to do the research work that will show you here. And this is again at room temperature, so there's nothing done here, just in water. The particles freely diffusing around. And again, I think the main message here is twofold, that at this length scale, everything is in motion, nothing is ever at rest. Yeah? So you always have to deal with and think about when you see a static picture that in fact everything is moving around, and more or less, of course, violently, depending on how much you're connected, but anything that is like a protein or a DNA molecule is really moving around a lot. So you always have to deal with these kind of issues with all kinds of sensing applications. Yeah? And this is, of course, a fundamental issue for all kinds of detection applications, because you always will have to deal with this kind of randomness in the system, and the fluctuations will become important, and I will explain where this is coming from during this talk. What's the overview? So I will start with basically setting out the challenge that we set ourselves last year when the lockdown started. I was quite surprised by the lockdown, but then we quickly said, okay, can we maybe do something with our technology for doing something useful? Then we could go back into the lab, and now we just filed a patent on this, by the way. Then I will introduce DNA nanotechnology, that I think is really a foundational technology on the nanoscale and molecular scale that is a bit larger than chemistry. Since we have the chemistry department, I assume that maybe a few chemists are here. But this is basically also on the intermediate scale, because molecular technology, but it's something that a physicist like me can understand. Then I will show a little bit about nanopore sensing and then combine everything together. And the two research sections at the end of the talk will be basically the single dynamic molecular dynamics in nanopore that we can measure directly by using DNA nanotechnology and nanopore sensing together. And this is really the basis for the detection and identification of SARS-CoV-2 virus sequences. And I will also show you that the technology that we develop is not just for SARS-CoV-2, but for any virus. Yeah? So we really didn't want to just do something that's just relevant for viruses, because arguably maybe we are coming out of the pandemic now, so we're a bit late for that. But we really wanted to have something that might be actually useful for future pandemics. Okay. So I talk a lot because I'm a physicist. I think it's always a good idea to talk about length scales and just make sure that everybody's on the same page. So if you have, we have two challenges in viruses. Yeah, that if you look at a human like us, it's roughly 2 meter length scales. If you, by the way, this is from the plug that was on the Voyager that was sent out to outer space and I think left the solar system a while ago. Then if you go 100,000 times smaller, you have a typical cell with a diameter of around 20 micrometers, so again 100,000 times smaller. And because you have this kind of different length scales, you see why you need a microscope normally to see a human cell. And if you go 10,000 times smaller, you actually end up at the molecule that we will talk about later much more, as DNA. So this is a photograph of the famous model of that Watson encrypted. It's a model of the model, of course, that you can see in the Cavendish Lab museum. If you ever can visit there again, if access is easier. So you can see here the double helix, and I will talk about this more, but the most important thing here is that the diameter of this double helix is around 2 nanometers. And again, the challenge here is that if you see here that you have this huge mismatch of 9 orders of magnitude, and again, normally you have it only in the astrophysical length scale, so you really can, there's a lot of room at the bottom, as Richard Feynman always said. And just to give you an impression here that SARS-CoV-2 is somewhere in between here. This is a rendering of the virus from electron microscopy. You see that the diameter of this virus is around 150 nanometers. The interesting thing is, of course, that all these systems are a bit different. They are not all 150 nanometers. it's like all cells are 20 micrometers in diameter, and these are just guidelines, whereas DNA, of course, has always roughly this diameter of 2 nanometers. So the question that we want to tackling now this evening is how can we detect SARS-CoV-2 viruses with high specificity and sensitivity. So the question is that this is a challenge, and the solution for challenge of high specificity will be found in DNA, because I will show you how we can design DNA molecules that bind to specific sequences in the viral genome that can identify the genetic information of the virus. And the solution for the high sensitivity is that we will try to analyze DNA molecules one by one, so we will really go to try to attempt to detect single molecules that have high sensitivity, and this will be achieved with nanopore sensing. And again, I will talk about this in more detail in the following. Let me just start here, this is evening talk, with a reminder of the structure of DNA. This is the famous sequence of life that is, of course, very nicely to see in the eagle pup with James Watson and Francis Crick photograph here. You see here the real scale of the model, and the physics museum actually the model is only this small because they lost the original model, or maybe the LMB stole it because they have a big model there in the atrium. When you come in there, the laboratory of molecular biology, and of course in 1953 they published a famous paper on DNA, and this is a famous sentence that I really like in this kind of way, and this I think what they got the Nobel Prize for, not the double helix, but actually this, that has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what are they talking about? And this is just, again, a quick reminder about DNA and the genetic code, that there were these 4 bases that were identified quite early on already in DNA are present. That you have adenine, thymine, guanine, and cytosine, so A, T, G, and C. And then you have this phosphate backbone that once makes this kind of double helical molecule negatively charged, because DNA is of course an acid, so the phosphate backbone is deprotonated in aqueous solutions at the usual pHs that we are working on, and then we have a negative charge here along the backbone. But what I want to focus here now is that A binds to T and G binds to C, and this is this copying mechanism that is kind of the basis for all inheritance in biological systems. And again, you see here the backbone running across each other, so we have this famous double helical structure, the bDNA form, and here you see the bases holding it together. And what we will see that you can, if you understand this system, you can also actually build things with DNA. And again, just another reminder what the length scale we are looking here is 2 nanometers across, so this is the rough diameter of this DNA molecule, and this is 2 times 10 to the minus 9 meters. Yeah, and it's a 0 with a dot and a lot of zeros behind. Yeah, it's really a small molecule that is not possible to see by eye. So now I want to just give you a quick glimpse of why this development is so important, because I think that the realization that you have this kind of base pairing is not only allows you to copy genetic material, but you can also use it as a building material. Because if you know that A always binds to T and G always binds to C, and if I say always, yeah, in most cases with the Watson-Crick hydrogen base pairing, you can almost see this that the DNA might be regarded as Lego bricks, but the special Lego bricks that only bind to certain parts of the other Lego bricks. Yeah, let me give you an example. So there's a famous structure that's called the Heavy-Duty Junction, that is a 4-way junction of DNA molecules. It's actually known for a long time that this can happen. So you can make a 2D structure that connects 4 double helices of DNA. Yeah, so if you make the right sequence, you can make a blue molecule, a red, a yellow one, a red, and a green. And this can also be found in nature for DNA repair. But now we can actually synthesize these 4 molecules. We can just call a company, they send us the molecules, and then we can mix them together, and they'll form a cross like this, like shown here. So again, the blue molecule binds to the green one and the yellow one, the yellow to the blue and red, and so you can build these kind of structures in this way. And the great thing about this is, if you understand this again, you can design, you can now go to a computer program cell, tell the computer program I want to have a structure like this, yeah, it will calculate the sequence for you. You can order this at the company website, and then within a week you can make billions and billions of these structures. Yeah, so it's really like molecular cooking. So it's mixing, heating up, and cooling down yields the design structures, because the lowest energy state in this mixture will be this shape. And I thought I might bring a video. You see it once, yes. So it's not a perfect example because this is only a diagram environment here, but here people from university in the US made this nice video where they had this shaped glass, yeah, where they added these kind of 3D printed designs, plastic systems with magnets inside, yeah, and then they provide energy, what would be in the molecular cooking just heating up. So again, we are back to the importance of fluctuation. What you see here, that after enough shaking, yes, this kind of unordered things develop into this nice. Let me just run this again, because I think this is really the key thing here, that you really can just add these kind of particles, and this DNA on the molecular scale it will be exactly the same. You can just add these molecules together, you shake them a bit, yeah, or a bit longer and a bit more violently, but in the end you will end up with a design structure, yeah, that's almost a perfect fit. And you see here that you might sometimes have to apply a bit more energy, yeah, shake it a bit longer, but fundamentally you end up with this tetrahedron in the design. And on the molecular scale, this was realized in 2006 by Prof, who was actually a computer scientist who came up with this idea to use DNA as a building material in this way. So he designed the system, the smiley face, yeah, this is, so what you see here, this thick line here is a double-stranded DNA molecule. So if you zoom into here, so this flat part is actually the DNA molecules that I showed you before, that there are all these helicate junctions, there's this four-way junction that keep it together. Yeah, you see here that the red strand connects this part of the strand with a light blue one, and we have the next junction here, another red strand that connects with the green strand, and so on. Yeah? So you can really build these kind of systems in a computer and make it. Then what we do in my lab, we order these kind of all these molecules together. This can be a few hundred. We mix them all together, put them into a fancy cooking machine. So this is a thermocycle that, or if there are some biochemists here, this is what we use for PCR polymerase chain reaction system. Yeah, you just put it in, heat it up, cool it down, and then if you take it out, this is now a microscopy image of this famous smiley that was on the cover of Nature in 2006. That actually shows, yeah, this nice way that you can build these kind of systems for DNA. And I hope if you see that you can make a smiley, yeah, it's probably not so hard to imagine that you can make any shape that you want. Yeah, you can people make now large structures, small structures. Whatever 300 shape you want, they make buckyballs, tentacular structure, everything is possible in this kind of system. And again, the reminder here that this is really like architecture on the nanoscale, because the building system, so the size of the smiley here is around 50 nanometers in size, and you really are having control over the three-dimensional shape of DNA molecules by just ordering these things together and cooking them. So it's perfect for experiments where you really need control over the system. And the other great thing, and this is something that I can't emphasize enough, that this is all self-assembly. So all what you do is you mix everything together. So the main energy is in the synthesis, and then you have to heat everything up, and then you make billions and billions of near identical structures in this way. Yeah, so it's really like cooking. So if you have a pot of this material, you just mix everything together, you heat it up and cool it down, and then you make billions and billions of these systems in parallel. And this, just as a comment here, this you can do in your lab at room temperature. And if you think about the number of transistors in your smartphone, yeah, you need a lot of years, yeah, to make billions and billions of transistors. Yeah, so nature is still better in making structures at this length scale fast and in large scale. And in a way, I always think it's nice that also DNA can be heavy. But let's go to the next one. So now we have the SARS-CoV-2 virus that probably made most of us very unhappy, because I couldn't leave this country for almost a year. So I was just in Germany last year, so I was very unhappy about that. But this was the bugger that led to all these kind of lockdowns. And if you look at this, It also is a kind of, with life again, it's a different discussion that maybe we shouldn't start here. But fundamentally what is in this kind of shell is the genetic information, and if you look in the internet you can find that the genome of SARS-CoV-2 is actually an RNA virus, so it's just a small change in the chemistry of the backbone, and I don't want to go into details here like this, but basically what you can see here that in the small virus shell, hidden is the viral genome. If you roll this up, this would be the predicted self-assembled structure of the genome. So you have a lot of, you have I think 30,000 bases or so in this kind of thing. And what we now want to do is we want to actually detect this, yeah. And what we will use for this detection is that we will. The nice thing about this is that RNA and DNA are extremely compatible molecules, so you can actually use DNA or RNA to bind DNA and RNA together. And this is shown here in this graph, where you can see that if you have a DNA molecule, an RNA molecule, and apologies, this is too small, but if you have now an RNA molecule with the right base pairing here, you can actually make an RNA molecule bind to a DNA molecule in a very specific position. Yeah? So it's exactly the same idea like with the self-assembly. If you design a DNA molecule with the right sequence, it will bind specifically to a certain position of the RNA. And if you now think that you have normally 4 letters, yeah, and if you have already, let's say you have 20 bases with 4 different letters, it gives you 10 to the 30 different configurations. So it's really, really highly specific if you pick the right sequence. Yeah? So you can really bind things highly specific to these kind of systems. Okay. So I can tell you now a funny story, because of course I'm a supervisor and who sometimes thinks he knows everything. So I was in January when I ignored all the reports from Wuhan, but then I had a very good and alert PhD student, Philip Boscovich, who said, wait for a moment, yeah, I think this will be a big one and we might end up in really big trouble. So he started thinking already in January, almost 2 years ago now, when this first happened, how can he maybe, the technique that he was working on, detect the RNA from the coronavirus. And he's of course, he's a very clever guy, yeah? So he had 2 motivations there. One is actually detecting the virus, but the second one was, if there's a lockdown, that we can claim we're working on coronavirus. Yeah? So it was both very clever and very shrewd. Okay, so and I will show you now where we got to from this. Yeah? So his idea was, again, I already gave the idea kind of away in the last slide, but basically the idea is because the DNA sequence is so specific, to find a place in the RNA virus that is highly specific and can identify the SARS-CoV-2 virus, cut a small of these pieces out, and then bind it to one of our DNA origami structures or DNA nanostructures that we have in the lab. And here I can show you an old paper. So we had this already a long time ago in 2013. We started to work on a system where we had kind of a DNA structure, and I don't want to go into too much detail here, but basically the idea was to present here different binding sites to a DNA molecule or an RNA molecule that we could detect from the level where it was binding, if it was actually binding or not binding to these kind of things. So then you had a kind of a crude sensor to measure the presence of this kind of sequence in this kind of system. So Philip knew this paper because he read most of the papers on the Nanopore sending in our group, but he also realized that this is not a perfect system, yeah, because you see that this paper is almost 10 years old. I'm a long time now in Cambridge, and nothing happened with it. Yeah, so it's there, it's cited, but we didn't really anything do with this because it's not working perfectly. So and then we had also a few years back, we had another idea how to make the world's most boring DNA structure. So basically what we took is we took a single-strand DNA molecule, and then Nick Bell at that time and I came up with the idea that maybe we can make a very simple structure with a lot of ends by just having short DNA oligos. Yeah, so again, this is not drawn to scale, so you have this orange DNA oligo always will bind to this position along the black strand, and the black strand is always the same. This is coming from a virus that infects bacteria, so it's very easy to produce. The pink one always binds to this position and so on, and if you mix them all together and heat them up and cool them down, you have a long molecule that is just double-stranded DNA. Yeah? First of all, it looks boring, but there's something interesting both from a sensing point of view and a physics point of view, because what you see here that we now have regular ends that are free to attach something. Yeah? So you see here that always there's a break, and this is also maybe shown here in this thing. You always see there's a break in the backbone of the colored strands. Yeah? And this gives you an ideal point where you can attach things to it. And Philip said, okay, maybe we should actually use this very simple structure and basically just attach, make this strand a bit longer, and this sequence, yeah, we will tune and to the SARS-CoV-2 sequence. So the idea would be that we have this long DNA molecule that we again call this DNA nanobase, and I will show this, mention this again and again in the remainder of the talk. But now, what you have to take away from the slide that we can actually make DNA molecules that have protrusions or overhangs of DNA here at certain positions where you can attach whatever we want. Yeah, so this is if you have a chemistry background, here you can attach here whatever. You can maybe also attach binding sites for proteins, for protein sensing. But in the rest of the talk, I will mainly talk about our ideas how to attach here something to detect the SARS-CoV-2 virus. So again, the ideas are extremely simple. Patent is filed now on this, so don't get any ideas and compete with us. But anyway, so the point is here we now have a very simple system where you can identify what binds here by knowing the position of your design. Yeah? So being absolutely concrete. Yeah? So if you know what sequence you positioned at the pink strand, yeah? If you now see something binding here, you know exactly what it is. Yeah? And the series, of course, then at the blue position, you can also see the same. So what you're combining now is, if you have this molecule, yeah, you can read out what is binding there at different positions. So now, of course, you can tell me, wait a moment, but how do I know? Because if you remember, the molecule is 2 nanometers across. How on earth can you actually detect what is bound there? And now we come to this thing, that this is something that started 20 years ago when I did my postdoc in Delft. So we were thinking about how to measure single DNA molecules with the aim of DNA sequencing. We never achieved DNA sequencing, but we got some other results that I will show you now a little bit. Yeah, so again, this was me doing my postdoc, trying to fill DNA into a small hole, and this is a typical hole that you can see here. This is a transmission electron microscope image of a nanopore, and this is really nothing else than a hole in a membrane. Yeah? I think nanopore sounds better than nano hole, so this is why we call it nanopore, and the diameter is 10 nanometers, but to be a bit more serious here, what you can see that this is basically roughly on the scale of a DNA molecule, yes? So a DNA molecule, as you can maybe remember, has a 2 nanometer diameter, so it nicely fits through here, and if you bind something to the DNA molecule at a certain position, you might be able to detect this in this way. So how does the detection work? And this is another extremely simple idea. It was so simple that in the beginning, it was actually the person who invented this principle tried to patent it in 1953, I think, almost the same year as DNA structure was discovered. The idea was so simple that the patent office didn't like this invention. Yeah? But let me show. So in the end, he patented the method, but the idea is very simple. Yeah? So again, we have this membrane here. Now we see a cut through here in this schematic. So you have the thickness of the membrane is L, the diameter of the nanopore is D, and you have it in the membrane, and here is the nanopore. And what you see or what you don't see is that it's all immersed in water. So we use water-based solvents, and then we add just table salt, or we don't use table salt, but actually lithium chloride. So the positive ions are lithium ions, and the negative chloride ions, and we apply voltage over this membrane, and then the current only can flow through this nanopore. I hope what is not hard to understand is that if you now put something through this pore, you will change the ionic current. Yeah? So anything that goes through this volume here that I showed in red can be detected and analyzed by just doing a simple ionic current measurement. Yeah? So it's a label-free sensor because you don't have to do anything to the molecule. You just pass it through this volume, and then you can read off the structure by just doing ionic current measurement. And maybe you know that in your phones, yes, the bandwidth of these kind of ARM processors that are running most of the smartphones have several gigahertz bandwidth now, so current measurements are extremely quick. Yeah? So it's extremely fast, so electronics always has a huge advantage for readout of biosamples. Yeah? And the other important thing here that basically this idea is just that we count ionic currents, count the number of ions that are going through this kind of sampling volume as a function of time, so anything that we can detect in here that changes the number of ions at this given number of time we can detect. So now again we come back to the fiddling part. Yeah, so of course now if you can imagine, so here's a nanopore as a tip with a diameter of 10 nanometers, so you can also make nanopores that look a bit funny. These are the glass nanopores that I mentioned before, and again you can imagine that fiddling molecule, if it's moving into this nanopore, is a bit different, so this is not how we do it, but what we're actually doing is using a trick. So let me just see if I can run this video. So what you will see in this video is there's a nanopore located somewhere here in the image. So there, of course, you can't see it because this is a microscopic fluorescent imaging, and these white dots that are dancing around are actually DNA molecules. Yeah? So of course, what we are not doing, we are trying not to manually take one DNA molecule and try to fit it into a eye of the needle of a nanopore. But what we are doing, we are just using diffusion and fluctuations and randomness and random walks. You can see here the DNA molecules are diffusing around. We have a positive voltage applied here, and maybe if you look closely, I hope the contrast is good enough. Maybe here it's actually better on this projector. You can see that some of the molecules are actually disappearing into this kind of glass channel that you're seeing here. And it's a tip again. There's a nanopore that is 15 nanometers across. These molecules are now fluorescently labeled, so normally you wouldn't do that, yeah, but just for the visualization of this technology, this is how it works. Yeah? So in a way, this is ideal. Yes, we can be now happy because actually fiddling the DNA into these holes is quite easy now. Yeah? You just have to wait. You apply voltage, you add enough molecules, you have to not make, of course. If you have an infinitely large reservoir with one molecule, this sensor will not work. Yeah? So you need to have enough copies of the molecules to actually get enough events per second, but you can already see here, this is again a real-time video of this kind of sensing thing that you can see that you get enough events in these type of systems. Good, so to recap, yeah, so I showed you now how we detect, and now I can finally show you the idea of the sensing principle again. Yeah, so we will have a DNA structure that we've shaped to detect the right sequences of RNA. Of the RNA virus, and we are doing this in a following way. Again, we have this kind of sensing volume that's shown in blue here as the outline, and then what we have, we apply voltage, we drive the DNA molecule through, and then you see these red balls here that are just highlighting the sequence where you can bind to, and here there are 4 balls located along in this cartoon, and then what you see that our signal will look like this. So this is the current flowing through this volume. The DNA comes in, so this is the first level, this is the double-stranded DNA, and then you see 4 peaks here that are the spikes down. It's called downward spikes here, yeah, indicate the binding of these kind of target sequences. Yeah. So the great thing about this again is that because we also always design these carriers of these DNA molecules with an asymmetry, you can always in principle say which molecules where. As you can see, we have here one is very close to the edge. Then we have a larger gap, and then we have a bit larger gap at the other side of the molecule, so you can also tell if the molecule is going forward or backwards through the nanopore because we don't have control over that. Yeah, but fundamentally, we can now, if we can now measure the position of these kind of spikes with high enough resolution, we should be able to identify anything that is carried through the pore with this DNA system. Yeah. So this is why, and the carrying is very similar because I think it's a very good idea to call it like this in the beginning, so we call it DNA carrier system in this kind of way. Okay, so I told you now what we want to do. Yeah, there's of course one problem here, and if you remember the first video, and of course in the real system it's not like this. Yeah, so if you remember the video of the fluctuating DNA molecules, everything is a bit more in motion. So the question is now, can we really identify which viral fragments we have along the DNA with localization of the binding site, and does the velocity change during translocation through the nanopore? And this is something that we answered with Kai Chen and some other people. And I would now quickly go to a little bit of physics. Okay. So physics, I think in my area, is all about time scales. Yeah, because all these time scales matter in these kind of systems. And so if you look at this is a nanopore with a DNA molecule that is just about to be pulled through. Yeah, this is a DNA molecule that would be the DNA carrier. And now it's of course surrounded by ions and water molecules that are constantly moving around because of the thermal energy. Yeah? So these kind of red arrows that you see here are supposed to be thermal forces, and they are constantly bouncing around the molecule and they give it this kind of curly shape. Yeah? So it basically dances around because of that. And now comes the interesting part here for the system. So I didn't tell you until now how quickly the molecule goes through, but it actually only takes a few milliseconds to pull a long DNA molecule that can be micrometers long through one of these nanopores. And now comes the question is, what is the time scale for this molecule to actually relax its position? Yeah? And if you want to calculate this and get an idea about this is the so-called Zinn relaxation time. And this is basically the time if you stretch out a molecule and let it go, how long it takes to reach an equilibrium configuration. Yeah? So this is basically scaling with the length of the molecule, and as a length, L0 is the Kuhn length, so this is the bond-like chain molecule, so the segment length, so this is the square root, scaling with the square root, or actually with square root to the power of 3, so 3 to the power of 2. And then you have the viscosity of the solution here, theta, and you divide by the thermal energy. So kBT, as soon as you see kBT in any equation, it's always entropy that plays a role, and this is a Zinn relaxation time. And then now we can do this in experimental systems that are published in the literature. We can now compare the Zinn relaxation time to the translocation time. And what you see here, so this is the translocation time or transport time is a function of DNA length. And this is the time where you have quasi-equilibrium, where you pull this chain so slowly that it can also reach equilibrium configuration. But you see that in most of these kind of systems that we are working in, we are in a non-equilibrium process, and non-equilibrium for physicists is always bad news, yeah, because most models break down, yeah. Because physics always depends on if you have a nice model, yeah, if that you want to have analytical solutions. Mixture of time scales or non-equilibrium is never a good idea, yeah, because then modeling anything is quite hard. Okay, so this is bad news. It's good news for experimentalists like me, because then you can do something and tell the theoreticians that they're not good enough. But then they tell me that I'm not good enough in doing my experiments, of course, but this is just the banter that actually drives this thing forward. Okay, so quickly now, just onto the physics. So what we did now, we made out a DNA carrier molecule that has these kind of markers. So we now set out to measure the velocity that the molecule has during the translocation. So we look at really, it's like almost like if you have an ideal gas molecule that you ask, okay, in an ideal gas, what does this molecule do as a function of time? Yeah, so this is what we can answer here, because each of the DNA molecules now will be labeled by these red markers that is just shown here. It's just DNA structure that we positioned around here, and then now we can know exactly the distance between each of these markers. And then if you measure the time between each of these spikes, you can actually measure the velocity by just dividing this time by this distance by the time when you see the different spikes. Yeah? So you can really, on a single molecule basis, you can, for a single DNA molecule, you find the velocity fluctuation in these kind of things. I don't want to bore you too much with the details. So these are high resolution measurements. There is different kind of nanopores. So you see again here, this is 100 microseconds. The scale bar, this is 200 picoamperes on the y-axis. So we really have to measure small currents very quickly, and this is not so easy. So what we did now, we wanted to see how the velocity changes. And you see here that this is a molecule. Again, these are markers. And to make it our life easier, we just added an end marker as well. So we have 7 markers now, not 6 like in the molecule before. And then if you look at the signal here, this is the signal as a function of time. Molecule comes in, you see 1, 2, 3, 4, 5, 6, 7 peaks. And by the way, if you ask me how good our assembly is, it's actually 99% of the molecules are correctly assembled. Yeah? Why? Because DNA self-assembly just works. Yeah? It's just good if you don't make mistakes with pipetting or putting something in. The other thing what I directly want you to look at, and what you probably notice, is that the different times between these markers is directly getting shorter, yes, or changing at least. Yeah, so you directly, I hope that tau 1 is actually smaller than tau 4, yeah, and tau 6 looks much shorter than even tau 1. Yeah, so you see that the velocity of the molecule is not constant during the translocation, and this is of course bad news, yeah, because if you now want to have a lot of markers here along, yeah, you might actually risk things. Or if something is not bound here, if one peak is missing, yeah, for a sampling application, you might want to know that, because then you need to correct for the sampling that a peak is missing. Yeah. To put this together, yeah, this is a measurement over average over thousands of events in 5 different nanopores. So if you now normalize tau 1, this is the distance between the first two spikes, if you now normalize the difference between two spikes, tau I, yeah, this would be tau 1. So the 1, 2, 3, 4, 5, 6 different time scales, yes, so we have 6 different I's. When you normalize this tau 1, you see that the molecule gets smaller, yeah, and it gets slower, so time increases, and then suddenly at the end it gets much faster. So it's a kind of a, so we can, and the interesting part is, despite the fact that all these 4 or 5 different nanopores are different sizes, we see that all of them with an error bars behave almost the same, and they all slow down around up to 10%, and then at the end there's a fast speed up of the translocation. And since they are all collapsing on the same curve here, you can hope that you really deal something with the polymer physics, so it's not the nanopores that are playing the role. Yeah? So you really see it slowing down during the event and speeding up at the end. And we really believe, because this is kind of an ensemble measurement by averaging over single molecules, because you can really get the ensemble averaging by averaging over thousands of these molecules, and you can see that they all roughly agree. Yeah? So statistical mechanics works. Yeah? There's unfortunately no surprises there, so the people who work on statistical mechanics knew what they were doing. So again, there's a slowdown and a speed up in the systems. But again, I just wanted to show you maybe. And then I told you already, analytical models are out of the question here, so we teamed up with a simulation group in Amherst in the US, and they did a simulation that we've shown here. So they actually took a similar idea to what we had. So they made our channel-like nanopore. Let me just run this again, maybe. Yeah, so you see these markers along this molecule, and you see that the molecule is actually dancing around quite a lot. So again, fluctuations matter. And you also see that the molecule is not relaxing itself, yeah, because you see that this will be a more relaxed confirmation in these kind of time scales. To look at this again, yeah, so let me just go over this quickly. So you see exactly in the simulation, you see exactly roughly qualitatively the same behavior. So the molecule first gets slower and then gets faster. Okay, why is that? And this is easy to demonstrate. Yes, so what you have here, you have the molecule here, and you start pulling. Yes, you can pull very slowly, then nothing happens, but if you pull very fast, it takes a while until it really starts moving. Yeah? So if you pull very fast and doesn't give it time to relax, you see that at the end here is kind of, well, it's chosen cartoon. You see that the end almost doesn't move, yeah, it only starts moving after you pull it very, very taut. So this is why actually the molecule is slower, because only then the whole molecule starts moving after a while, but then if you retract it, because all the friction on the molecule side here is important, and it actually speeds up much faster. And I think the interesting thing here is really that the understanding and quantification of this velocity profile is really general and can be used to correct for position of the markers in these kind of measurements. And just to maybe show this again, yeah, so the other important thing what you can also learn here that depending on how the molecule is actually shaped before it starts its translocation, it actually changes the speed, the total translocation time. Yeah? So it also shows you that on these systems you don't have a very, so the distributions are really reflecting conformations, molecular conformations that you can directly have access now in the experimental data. Yeah? So just to show you, this is from the simulation, of course not in the experiments. So if you have a coiled-up molecule, you find that this is actually going through quite fast. And you have a medium where you have some coils and some straight part, and this is kind of going through the average. This is the most common translocation time, and then if you have these very stretched out molecules, they are much faster, slower. Yes, and in this kind of race here, in these two classifications, it's hard to see, but maybe you can see that actually the top one is actually faster through the pore, and the end is faster to the end, yes, than the lower one that is stretched out in the beginning. And again, this makes complete sense, because if you have your molecule stretched out in this way, yeah, completely stretched out, and you pull directly, everything starts moving. Yeah? So it makes absolute sense in these kind of systems. So again, with these two regimes, now we can actually start to maybe detect coronavirus, yeah, because we can now look at and correct our marker position if we want to with this knowledge of these kind of systems. And again, this is a general feature of all these kind of nanopore systems that we are looking at. The linear slows down, and then speeds up, and the correction of these velocities allows us to identify the markers. Okay. Good. So now can we do this? So again, we just go this over quickly. Yeah. So in the human cell we have DNA. It's blue shown in here. We have SARS-CoV-2 viruses. We have RNA molecules. And usually, what you do, and I don't know who did a COVID test. Do you all in college? They probably do all COVID tests, all swab tests. Yes. Okay. So do you know what the college does? Do they still do PCR? Yes. So this is of course a typical standard test. There you have the swabs, then you do the lysis buffer, you get the RNA extraction, and then what you have to do in this kind of system, you do copying and amplification. Yeah? So what you can't do, you can't directly detect the RNA in these systems, but what you do, you take an enzyme, an RNA polymerase, it translates the RNA into DNA, and then the DNA is actually amplified. Yeah? This is what they call the CT values, that maybe people who have positive PCR tests may know that, what the CT value is. And then after copying and amplification, you can detect this. And this is costly because it uses enzymes, and it's kind of slow, yeah, and it's not an ideal technique, yeah, because again, these PCR reactions take time and are also, so we want to replace that with our technology. Yeah, we want to directly measure the RNA. We don't want to go through this thing. So, of course, the PCR tests are really good if you want to detect very low quantities of virus, or very early detection, or avoid non-asymptotic transmission. We are not interested in this. We really want to detect directly the RNA, and I already told you how we will do this. Okay. So then, of course, we said, okay, coronavirus is probably going down, and we actually want to have a technique that can also be maybe used for future pandemics, and not only that, but also maybe useful for doctors, because one problem with respiratory symptoms is that if you have a cough or a sneeze or some kind of runny nose, you are not sure which one of the viruses it is. Yeah? And sometimes you want to know. Yeah? So sometimes also for treating the viruses, you want to know if it's called, is it SARS-CoV-2, is it an influenza virus, rhinovirus, RSV, and para-influenza, and all of these viruses, the nice thing for us is that they are all RNA viruses. Yeah? And the other important thing, what you also is even worse in the whole situation, that if you have a lung infection, so pneumonia or something like this, but the other important thing is that you also want to see if it's a viral infection or a bacteria, yes, because then you can only decide what the correct intervention is that you want to do. Yeah? So you really, in principle, want to detect all these viruses in parallel, and this is something that at the moment is extremely difficult and costly to do with any of the techniques. So what we set out to do with Philip, so Philip designed these kind of, looked at all these genomes, and decided which parts of the genome we can cut out to identify this kind of system. So we designed now, this is now a very simplified way of our DNA carrier system. Yes, we have 5 binding sites, and each of these sequences defines one virus. Yeah, so we have one for rhinovirus, para-influenza, RSV, influenza, and SARS-CoV-2, so that all on one thing. So if you show up in the hospital, they can tell you directly, hopefully in the future, what you have. So we designed the system, so this is how it looks like. Yeah, so we have a binding site here, and we have a blocking strand. This is a bit, I don't want to go into too much detail here, but we are trying to enhance the signal here, and we have a negative test where the signal disappears when we are detecting the virus. The presence of the viruses has some advantages, because then you can make sure that the test also worked. Okay. So again, so this is an atomic force microscopy image of one of our molecules here. You see that this is the DNA molecule, and then hopefully you can see these kind of small protrusions here. And again, I'm sorry for the low on the screen, it looks a bit better. And you see these white blobs, I hope, and these are these kind of markers where these DNA and RNA strands of the viruses can bind. Yeah? And if the probes are absent, this means we have a positive test. So this should be positive, of course. So the test is positive if the probes are absent. So this is a reference structure here. It just tells us it makes the whole system asymmetric and helps us with the readout of the velocity fluctuations. And I hope you can see here that here there are no white dots left on this kind of molecule. But again, so what we do here, if the virus is present, these black dots disappear, or these white dots here on this graph, and then you have a positive virus sample. So I hope I now didn't mess up the slide. So the probes are present, this is negative, so this is how a typical event looks like. So this is a DNA molecule going through, so this is the level of the double-stranded DNA. The DNA carrier goes in, or the nanobate. And then you have this first peak, that is the reference structure that we have, it's all DNA, and then you have 5 peaks, 1, 2, 3, 4, 5. You see actually this is a typical event where not all the peaks look the same because we have some challenges there with the molecule moving not through in a constant velocity, but basically in 5 peaks, and then you have the end peak here that tells us when we have to stop reading. So now what we can do, we can actually add now it's correct, luckily, so if all the probes are absent, means that the virus sample would be positive, and this is just an example where we added DNA to see that all the probes work. So Philip just added all the 5 targets to this kind of sample, and then we see that all the peaks are absent. So we can directly see from our system, here we don't have viruses, here you have all the virus sequences present. So I have to be accurate here. This is not, of course, a virus, it's really, we don't detect intact viruses. We always need the RNA, so the RNA needs to be extracted from the lysis buffer, so this is very similar to the PCR machines. But fundamentally, you can see that it's very easy to see in our system. This is a single molecule detection method. You analyze one of these nanobates, and if the peaks are gone, you know which virus is present. So again, to show that this is really working, so we did this now with these five different viruses. We built this kind of molecule. Now, maybe if you add RSV virus, you would expect that this one binds to this position, so it removes this kind of peak. This is exactly what you get. If you look at here, this is a typical event in our nanopores. You see here that this is plus the RSV virus, and you see that the orange peak is missing. So with no target, you have all 5 peaks present, and then with the RSV target, you have 1 peak missing. So the great thing about this is great signal to noise ratio, and you can detect these kind of things. And you can, of course, add rhinovirus target, influenza A target. You see that then the 3rd peak is missing for the rhinovirus, and the influenza A target is missing, parainfluenza, and you can really go through this kind of work. And then if you look at this kind of thing, so the higher the peaks, the better the detection. So we call this displacement efficiency again because we say we are having a molecular displacement here at this position, and then you see that SARS-CoV-2, RSV, rhinovirus, influenza A, and parainfluenza all can be easily detected. And this all can be done in parallel, and in principle we could also do more viruses because we could also have more binding sites here on these molecules. We could also have several of these molecules and design different ones, and we could maybe make a test with 50 to 100 viruses in parallel. That can be all done relatively quickly. Okay. The other interesting thing is just to show that we can also detect variants. You probably heard all of this. That we are all the people who are now getting infected in the UK and got vaccinated. I had coronavirus 4 weeks ago. Despite double vaccination, but I had a very mild case. I'm not even sure if I had it, but I definitely had a positive test and had to self-isolate. But basically, this is probably all just the different variants. This is the nomenclature that they work in our technique can even detect different coronavirus variants. So it's exactly the same idea. So I don't want to go bore you with the exact sequence details, but just say that you can, this is technology, you can really detect even single base pair mismatches in these kind of systems because we use this, because the DNA RNA binding is so specific that you can really pick this up. Yeah? So if you have no target, again, we have all 5 peaks present, then if you have at one reference, it only displaces one signal, then if you displace the other one, you see that the second peak is missing, B1617, B1, B117, B135. Yeah? And the delta variant is actually the one that is at the moment, I think, in the UK, highly prevalent. Yeah? This is actually the yellow one. And so you see that, again, this is displacement efficiency. The higher the bar, the better it works. But you can see that it's highly significant. Yeah, you can easily detect this in this way. Of course, it's not perfect. We still have to do concentration detection measurements, but this is basically what is possible. So what I showed you so far was all the synthetic RNA, so it was just development, but now I want to just show you that, so now we have a collaboration with Steve Baker, who's at the medical school here in Cambridge, and he gave us a sample of a patient who was was during the height of the infection, so 3 to 4 days after first symptoms. 3 to 4 days, but just when the first symptoms came up, and I think the highest viral load is. So what we did is, so he took a nasal swab, yeah, did the usual extraction. So we have this total RNA extraction. So after the lysis buffer, then we do our extraction kit. So we have this RNA H cutting. If you have questions about this, I don't want to go into detail, but we just use a clever enzyme that nature gives us that allows us to cut out the target sequence. We do this, then we mix it with our DNA nanobate, yeah, that has now 3 target sites. And if you do this, we have a negative patient sample where you see that all the 3 peaks are present. And in the positive patient sample that we did in parallel, you see that some of these peaks are missing. Yeah? So why are only not all of them displaced when we have a patient sample that has a high viral load? Because our method is, of course, as I told you, not for any detection of the low things, but actually works for when people are highly infected. And so we envision that maybe it can be used in the hospital setting and people need to decide, okay, do you have corona and go to isolation, or do you have another virus and or even a bacterial infection, and then decide this very quickly. Yeah, so again, but this is all single molecules, readouts, yeah, these are just typical events, and what you can see that we can, at least in this example, we can definitely detect negative from a positive sample. So what is basically what you need to do. Yeah, so you can tell, okay, this person had the SARS-CoV-2 virus, and of course it also had COVID. Okay. Looking at the time, I think I will end here. So I gave you now an impression, I hope, that we did this single molecule biophysics, so we can quantify velocity fluctuations. So this is basically the physics, these kind of polymer physics on the molecular length scales, where we actually mark these kind of DNA molecules as markers, and we can really measure their velocity fluctuations at lower length scales. And one application I focused today in the talk here was actually detection of viruses. So this is, of course, different viruses, or we can go for virus variants. We can also do pooled sampling, so we can look at a lot of different patient samples at the same time. So with the DNA nanotechnology, we really have a lot of possibilities to maybe help something or do something useful in coming academics. Because the other thing what I maybe should say here, that it's really easy to adapt this test. There's all that you have to do here is you have to change this one DNA strand, yeah, in the system, and then you have to test for whatever virus you want. I didn't have time to really talk about the localization, despite the fact that I promised to talk about it a little bit, and I just want to say that we can indeed localize single binding sites along a DNA molecule with 7 nanometer resolution. That is done with a 5 nanometer nanopore. If you have questions about it, I'm happy to answer things about this. And then the third application we envision for all this kind of systems that is also related to the velocity fluctuations, actually data storage, because with DNA data storage you might be able to reduce the requirements for data centers in the future. Before I end, I would just like to acknowledge the key people from my lab who worked on this. Nick Bell was a PhD student and postdoc in the Microscopic Institute, who started with this kind of linear DNA structures. Philip Boscovich, I mentioned several times, came up with the idea. Like Chen worked on the velocity fluctuations, the postdoc. Nick Ehrman did the data analysis with some machine learning there. Alex Oman helped with the DNA structure design. Ran Jiboni did the AFM measurement, and Jimbo Zhu actually helped with the design of DNA systems. And then we have a very fruitful collaboration with Stephen Baker, who's actually here at the Cambridge Medical School in the university, who gave us samples and helped us a lot with insight of what needs to be done. We used materials from Mark Howarth's lab in Oxford, the DNA the DNA self-assembly we learned from Tim Beadle, and I told you already that in the USA we have Mugabe Puma who helped us with the simulation systems. Yeah, and I should say that part of the study, no, sorry, not of the study, part of my lab is actually funded by Oxford Nanopore Technologies, and Iridia is a DNA data storage company, and then we have some other sponsors that maybe don't know what we're doing, but maybe it's better like this. Okay, with this, I'd like to thank you for your attention.
Thank you so much for this wonderful talk, Professor Ulrich. Does anyone have any questions?
So I didn't quite catch how the probing DNA junctions were made. Like, were they nipped and peeled off, leaving a part of single-stranded DNA?
Yes, so all that we do here, you see this gray part, it's always the same, yeah, and then all that we do is we make a longer one, yes. So we just order a longer stable strand. So when it was before 38 bases, yeah, it's now 58, 50, 60, whatever. You can just buy it. So this is why it makes it so simple, yeah. Because you can just go to the company and say, at this position, I want to have a strand that is 60 bases long with this sequence, yeah, and then you have your binding site built in. So this makes it extremely easy to adapt to detect anything. I hope that helps. Yeah. So it's really it's super easy to design these things because you can order all these things from our company, yes, and you don't have to do we just build them, we design them in a computer, send the sequences to IDT or any other company who makes these oligos, and then you can have them in a couple of days, yeah, make these molecules and they are adapted. Yeah. So it's extremely easy to design it.
So are they more like annealing primers with overhangs?
Yes, yeah, so if you want to call it overhangs, it's also fine. Yeah? So you can also call them overhangs, yeah, whatever. I mean, it's just a single-strand unpaired basis. Just as a comment here, so we use these overhangs also to have a blocking strand here that is just partly blocking it we are doing this to enhance the sensitivity, because if you do this, if you have the bias sequence, it actually binds, it has to displace the strand, yeah, then you get the negative readout, yeah, this helps a little bit with the sensitivity. And actually allows us to see this peak going away, yeah, so this big white square is a protein, yeah, so it's a scapegoline that we get from the Howard's lab in Howard lab in Oxford.
Thank you.
Was there any other questions?
I have one question. I'm just wondering what are the properties of your probes that causes the signal to drop whenever it passes through the board?
Yes. Okay. Another picture. So we have different probes there. So we can use different probes. Yeah, so you can have DNA structure. So Jimbo Zhu, one of the postdocs, actually made some DNA structures that have this kind of. So you can make 4 helix bundles like this. Just double check where they are. Best images here. is now something. Okay, fine. So we basically, sorry, someone stop working. So what we do is we take a structure that can be a protein that has a molecular weight of 160 kilodalton or 60 kilodalton, depending on what we want. It's actually bound to this DNA strand. Yeah? So if you go back, okay, so basically it's a DNA strand. Sorry for that. We have to switch computers because it didn't work this morning. Yes. So this white structure here, yes, can be a DNA structure, can be a protein structure that makes it just an additional peak. Yes. So this is just a placeholder for whatever structure that has a high enough molecular weight that you can see easily in the nanopore. Yeah. So whatever you want to use there, so what we take care of that this is highly negatively charged. Maybe I should have said this before. Yeah? So all these molecules that we are using are always highly negatively charged because the glass surfaces of the nanopores that we are using are also highly negatively charged because we don't want things to stick on the nanopores. Yeah? So maybe you can just go back to that and emphasize again what you can nicely see here. Yeah? So what you see here in this kind of system, there's no charge here. So this is in the simulation. There's no, there's an electric field applied, but there's no charge on the system, and if you run it, yeah, you see that at the end, the DNA molecule actually sticks to the corner of the nanopore. Yeah? So, and this is something that probably, if this would happen in the experiment, it would directly end the experiment because then the DNA normally gets stuck. Yeah? So what we do, in the experimental system, we always have highly negatively charged surfaces. Yeah? So to use electrostatic repulsion to keep everything away. Yeah? And the black balls here that you can see here are normally proteins or something like that. Yeah, so that makes a piece.
Right. And how do you ensure that the viral genome displaces the probes on the nanobase?
Yes, so that is, okay. This is a good question. So what we do this by having enough of the viral target, yes. So this is why we are not very sensitive for very low levels. So we need, okay, this is an energy competition. Yeah. So what we do is we have a strand, yeah, that is overhang, and then here the viral strand bind, and then it displaces the other strand, yes. So if I would have a white, sorry, I didn't expect to have a whiteboard. So you basically use DNA strand displacement to actually push the probe off, yeah? And this is just, again, by using the lowest energy state will be that the probe actually, so that the target RNA binds to the molecule and displaces the rest, yeah? So you see here, in this graph, I think it's kind of shown. So what, no, this is not a good one. So this is also, it's not perfect actually now. So what we do here, so we have here this small overhang here, there the target binds and then it displaces, yeah? So if you have the, if you see here that the orange one is a bit longer than the gray one, yeah, that's blocking it. And this is enough energy difference to displace it, but this provides an energy barrier, so we need enough of the orange target strand, yeah? So we will not detect a single molecule, but we actually need probably 10, 50 more than of the target, yeah? So this is how it works. And it's a limitation, yeah, of the technique, because then this also means that we are not really super sensitive to low concentrations of the virus, yeah?
So in a live sample, you have like a mixture of a lot of different nucleotides. So does that cause a lot of noise in the experimental measurements?
Okay, luckily, okay. It does, but it's not a problem for us. I can tell you why. Okay, it's like, okay, so the great thing what we're doing here, we are shaping molecules that are, so we're looking for highly specific signals, yes? Mm-hmm. So even if you have other molecules that have different shapes or doing different things, we know that these are just background molecules that we don't care about, yeah? So you're absolutely right, yeah? In the swap, and this is what we try to show here, yes, you have all kinds of different molecules in the background, yes, that are not but they are all not looking like the system, that like the DNA nanobase that we designed. Yeah?
Yeah.
So we use the molecular design really to predict what structure you have, and only if you see that structure, yeah, you have the virus. Yeah. So this is really the key of this technology, that you really can have this kind of, so you really design in this foresight, yeah, what you want to have. Yeah. Otherwise it wouldn't work. Yeah. Because otherwise it would be just a zoo of stuff that you can't distinguish. Yeah.
Right. Thank you.
In terms of specificity and sensitivity, how does this detection compare with lateral flow testing?
Okay, okay. Yeah. So with lateral, so we are not there yet, but I can tell you numbers. Yeah. So what I can tell you that, so the effective concentration of RNA targets here in these kind of samples were probably 0.1 picomolar to 0.5 picomolar. Yeah? So this is the concentration of the target here. And this is already in a virus sample where we know that so we benchmark this with qPCR measurements, so we know that this was a patient with a very high viral load. Yes? So we know, yeah, that we can't detect people who are not at the height of the infection. Yeah? As we already know at the moment. Yeah? There are ways probably around this. There's a bit of sample preparation there to concentrate everything, but basically I think we can't outperform. So I don't think that this is the right method to compare with the lateral flow test as you do at home, but it's really more for somebody if you want to, if somebody comes with fever and pneumonia, yeah, and is really sick, that you check, okay, do they have a bacterium or do they have a viral infection? So this is why we believe that's can complete. So we don't believe that this is any method, yeah, that can replace lateral flow tests, yeah, because they are so cheap and so easy to do. Yeah, I think that's very hard to compete with. Yeah, that's not the idea. But it would be nice that we tried this in the beginning. But I think if you look at the technology, how good it is now, it's really difficult.
Do we have any other questions? If not, can we just give Dr. Ulrich another round of applause?